Hello, everyone, and welcome to the Statties 2020 Online DOE Summit, where you get your daily dose of DOE. Today, we've got a presentation entitled Deep Breath, Simultaneous and Quick Determination of Two Ingredients Concentrations in a Solution Using a UV Visual Spectroscopy Chemometric Model by Sam Guizani. That is a mouthful. But very descriptive. Let's see. Oh, I am sorry, I'm not sharing my screen right here, folks. And let's see. There we go. Okay, so there we go. And come on now. And here we go. A little about Sand. He's a process scientist at the Ferring International Center in Switzerland. And he's going to be talking about some of his current research and the model that he developed. And I'll let you read through some of that while I say at the end, we'll have some time for some questions and answers. And if you go ahead and type up your questions, you'll see in the menu on the right-hand side, there should be an area for questions, and go ahead and just type them up in there. And when we get to the Q&A section, I'll read them off to Sam and we'll answer them from there. And we'll get through as many questions as we can. So without further ado, I'm going to hand over the presentation. And
okay. Now, thank you, Greg, for this introduction. I think now I can share my screen, right? So I'll try to do it.
Whoops. Let's see.
Is it working?
We're seeing your. You should probably show the
presenter
other
view.
screen. Yeah.
Okay, I see. I got
there
it. So maybe this one is correct.
Oh, yep, perfect.
Good. Thank you. Thank you very much. So, welcome everyone to this presentation. I will be giving about the determination of the composition of two ingredients in solution, and we will be using UV-Vis spectroscopy combined with chemometric modeling here. I just want to take a pointer so that I can use it during the presentation. Okay, so I'm working for pharmaceutical industry, and maybe some of you are also working for pharma industry. And what is known in the pharma industry is that for the products we produce, we have a number of critical quality attributes. And among these critical quality attributes, there is the ingredient composition of the drug product. And being critical quality attributes, often we need to really test for each batch the content of ingredients we have in the final product. The product I will be talking about is a solution. So it's an aqueous solution with an active ingredient, API, in short, and excipients. The API concentration is about 0.1 milligram per milliliter. And as I said, it will be tested. It needs to be tested in the final product. The excipients, usually we are not required to test the composition in excipients in the final product. However, if these excipients have specific role, like for example, we have in this formulation, one of the excipients, which is a preservative, we need also to test it and make sure it is compliant with the specification. The reason is that this concentration will actually be determinant for the shelf life of the product. Currently, we test the concentration of these two ingredients in our formulation using liquid chromatography, HPLC, and we are using two distinct methods to achieve that. These methods are typically long lead time and typically offline methods. That means we produce the batch and then we pick up some samples from the production, and then we get the results after testing them in the QC lab. The manufacturing process that we apply for this product is a batch process, like many drugs in the pharma industry. However, we have a project where we wanted to switch from batch manufacturing to continuous manufacturing for this drug product. And here is a view on what the process would look like in a continuous manufacturing. So our API is stored in this device, which we called API microdosing, which will feed the line with grains of API, so small quantities of the API. We have also a feed of the line in terms of buffer solution through a pump that will ensure a constant flow of buffer into the solution, into the line, sorry. And in this device, we have a dissolution occurring where the API got dissolved into the buffer stream. It becomes obvious in this case that we will need a quick method to determine the API concentration in order to adjust on the fly the API flow rate and be within the specification at the end. And of course, if we collect the data over time, meaning the concentration over time of the product, then we will have no need actually to do it on the final product. We can substitute these measurements online to the measurement offline. In the same time, if we do the same for the preservative, then we will also avoid testing that concentration in the final product, and we can aim for real-time release of these products. During this project, it became clear for us that UV-Vis spectroscopy is a good approach to apply. And to show you that, I will show you the next slide here, which is actually the spectrum of the different components that we have in the solution. So the methionine here is the preservative that we are using, and the carbetocin is the API that we are using in this formulation. We have other ingredients as well into this formulation. But what we can see here is that we have an overlap of the API and excipients spectra. However, if we look at this area, about 230 to 310 nanometer, we can actually see some differences between the spectra that will probably be useful to quantify the amount of each ingredient in the solution. Of course, it is clear from this that a single wavelength classical approach would not do the job, and we need to turn to a slightly more complex approach, which is using multivariate analysis, in other words, applying chemometric methods. A few words about the analytical development approach we have followed. We have used in this approach QBD methodology, and you can say that we have used an analytical QBD approach, which combines systematic identification of factors that could affect the performance and the results of the methods or of the method, and also risk assessment to define where to put the effort, experimentally speaking, when we develop this method. Our development strategy has covered optimization of spectral acquisition parameter through using design of experiment. I won't detail too much this part of the development, because I think it's more interesting to focus on this part, which is about multivariate analysis, model calibration and validation. And during this, we have identified multiple factors that could affect the calibration and validation, like, for example, which range of concentration we do study, and how many replicates we include into the calibration, and many, many other factors that we took into account during the development. As I said, we will be focusing on the MVDA, multivariate analysis model calibration and validation. But if you want more details, you can go to this article that we have just published, so in June, in Pharmaceutical Development and Technology, where you will find all the details of the approach, besides the slides I will be presenting today. So first of all, let's look at the calibration and validation samples and how we selected them. Our design space is basically a two-dimension design space where we have the API on one side and we have the preservative on the other side. They can vary freely, so we can have any combination of API and preservative into this solution. However, design of experiment are known to really be very good tools to cover a design space. That's why we actually used a central composite design, a face-centered design in this case, to locate which combination we should create or which combination we should use in our calibration approach. These blue dots that you see here, we call them the calibration set number 1. And we actually performed this or got to these concentration, these combination of concentration using 6 stock solutions. Let me explain what is this about these 6 stock solution. It was impractical for us to independently prepare every single combination independently, because it implies weighing operation. It's actually quite tedious. It is more practical from an experimental point of view that we prepare stock solution which are highly dosed API concentration solution and highly dosed preservative concentration. Okay, and then we mix them in appropriate levels, appropriate amounts, sorry, and we dilute them also appropriately if needed, so that we can reach any point in the design space that we have here. Okay. The downside of this approach is that if we make a mistake, if we have an error while we are preparing the stock solution, then this error will be translated to all the samples that are manufactured using these stock solution. So to overcome this and to account for randomness of error while we prepare these samples, these stock solution, we actually did 6 stock solution for API and 6 stock solution for the preservative, and then we combined them randomly in the samples that we are using for the calibration. We added to this design what we called here the calibration set number 2. They are using the same stock solution, and you see they are located somehow in between some of the samples in the calibration. We will use them for evaluating the performance of the model that we create. And finally, we added another validation, another set of samples. We call it the validation set, which has been produced using freshly prepared stock solutions. So these would be samples that have nothing in common with the original blue samples or yellow samples here. They will be, for example, manufactured with a batch of API that has not been used for the rest, and we will use these samples validation to test the performance of the method and the model that we have created. A little bit about the transition from design of experiment to multivariate analysis. In design of experiment, the goal is to fit a model to predict a response Y based on some inputs X. In our case, what we would have done in design expert, we define the level of the component. These are our inputs, and we are measuring the spectra on the other side. And by the way, the spectra here is represented by a big table with more than 800 columns corresponding to the wavelengths we have in the spectrum. In our case, doing a regression of the spectra versus the component levels is of little interest. What we are interested in is to be able to predict the composition from the spectra. So that's why we have not used actually design expert in this situation to do this correlation. We have used design expert to define what would be the best combination to study, but we have not actually conducted the regression work in this software. What we will be doing, and I will show you that, is that we will be switching actually the position of the two matrices. So now our spectra will become the Y's and the input variables. We will still call it Y, but we have the responses, which are the component level. These are the output we want to measure. And we will do that using multivariate analysis regression in a software which is called Unscrambler. And Unscrambler is actually a partner of Statis, so this is why we are using both softwares. Okay. Why do we need to go multivariate in multivariate analysis in our case? I think there is two reasons for that. The first reason is what I mentioned about the number of variables that we have. Imagine you have here 800 or more than 800 columns. These are the variables that we use to predict the composition here. So imagine a model, a linear model, for example, with 800 variable, and you add to that all the interaction and all the quadratic terms, that would yield really a very complicated, let's say, polynomial model if you want to go to that direction. And the second reason is, I think, even more important in this case, is that we have a lot of correlation between the variable we have in the spectra. Imagine if you have two close wavelengths on the spectra, these the value that we get for the absorbance at these wavelengths are most likely very, very highly correlated. And we need to account for this correlation when we create the regression model. And this is what multivariate analysis is doing for us. I will be describing now two tools in multivariate analysis that we have used. The first one is called Principal Component Analysis, PCA. And actually, we will only use the spectral data. We will not use the information about the level of concentration of the component. So PCA is, for that reason, called unsupervised method. So we will only use spectra. We will not use the information about the content of the different component. It's a method that is used to reduce the dimensionality because it creates new latent variables that we can call also principal component that summarize the variation we have in the data. And finally, it is often used to do some clustering of the samples. If we apply PCA, so this algorithm, if we apply this method to the calibration set number 1, so you remember these are corresponding to the phase-centered design, we use only the spectral data and we apply no pretreatment, no spectral range selection, and we just look at what we got. So now we have a plot where each point here is corresponding to one of the samples, one of the measured spectrum we have in our data set. And I've colored them according to the replicates. So as you remember, we have 6 stock solution and we combine them randomly between the API and the preservative. And therefore we have 6 different combinations. What we can see is that these new coordinates, which are PC1 and PC2, are actually describing 64% and 32% of the variation we have in the spectra. And this is what I meant earlier by reducing the dimensionality. We started with a data set where we have more than 800 columns, and we summarized it using only 2 variables, so principal component 1 and principal component 2, which account for 96% of the variation we have in the spectra. So it's really a very interesting tool to reduce the number of variables to manipulate. What we see on this chart this plot, sorry, is that we have clustered the samples according to the replicates. So you have the blue replicates, the red replicates, the gray, blue, light blue, and green, and here a brown replicate, our 6 replicates. This is interesting, but actually we are more interested in seeing how we can actually map these variables that we create, the latent variable, the principal component, how we can map them to the composition of our samples. To do that, actually, we need to do some manipulation of the data. And what I mean by some manipulation of the data is that I will take the same data set, but instead of using the raw data, I will use the first derivative of the spectra, and I will use only part of the information I have in the spectra. I will only focus on the range between 240 and 300 nanometer. Remember from the raw spectra that I presented earlier, this is the region where we have, let's say, the maximum chances of being able to quantify each of the component in the solution. And this is what we get now. So as you see now, the replicates are clustered together. So these are the replicates that are close to each other. And we will see it later, but now we have a new pattern of distribution on this graph that is related to the composition of our samples. I will show it later in another slide, but before we go further, you can notice that we have one of our replicates, the blue one here, which is slightly different from the rest of the population. So we need first to, let's say, investigate or understand a little bit more about it. Here I'm plotting the same graph, and I've just highlighted the blue samples. And as you see, the blue samples seem to be simply similar to the other samples, but just shifted to the top left corner. And as I said, we can actually use another principal component. So now I'm plotting, there is a typo here, it should be PC3 instead of PC2. So now I'm using an additional principal component, and it is clear that this principal component 3 is only here to explain the difference between this sample set and the rest of the samples. So the most likely explanation of difference between this group and the rest is that something went wrong during the production of these stock solutions, the stock solution, and that affected all the samples of this sample set. So just for the sake of the demonstration, let's take it out and see how this affects the principal component analysis. Now the principal component analysis with the outlier replicate removed will make clear that the clustering is according to the component level. So this is the score plot where I have PC1 against PC2. So principal component 1 versus principal component 2. Principal component 1 is explaining 88% of the variation, and PC2 is explaining 12%. So together they are explaining almost 100% of the variation we have in the spectral data in our data set. And I have here only colored the plots according to the API concentration. And clearly what we see is that the API concentration is growing from the left to the top right corner of this graph. If I redo the same chart or the same graph, but now using the preservative concentration as a coloring indicator, then we see that we have another direction which is orthogonal to the first one, describing the preservative concentration. So that means using these PC1 and PC2, we are actually being to retrieve where are the samples, or to map from the spectral data and using PCA to which composition of sample we are looking at. And to convince you about this, I have here plotted the same graph using now the usual coding, which is the replicate. I've excluded one of them, which is an outlier, and therefore we have only 5 different colors on this graph. But as you see, it's exactly the structure of the design of experiment that we have created at the initially, with the API on one axis and the preservative on the other axis. So that was really very promising to see that only giving the spectral data to this algorithm, this principal component analysis, we could actually map to the concentration levels. Now I've taken all the sample I have, so that includes calibration set number 1, calibration set number 2, validation samples, and I've just recreated the PCA using first derivative and using 240 to 300 nanometer spectral range. I've also taken out the outlier that we discussed earlier. And as you see, again, we can rebuild the structure of the design of experiment, but we can also see that the calibration set 2 samples are falling exactly where they should be within that space. And the validation sample, as you remember, were produced using new stock solution. They also fall where they should be within the design space. So let's summarize what we have found with the principal component analysis. PCA was applied on the spectral data without using any information about the sample composition. And what we found out is that PCA allows to identify which pretreatment and which spectral range we should use that are most relevant to sample composition. One thing to really to highlight is that the DOE structure, in other words, the sample composition, is somehow encoded in the spectral data, and that PCA was able to rebuild this composition based on the spectral data, and that is using only the latent variable PC1 and PC2, for example. PCA was also a very valuable tool to identify outlier groups. And we have seen that it is linked to one of the experimental replicates that we have done. Let's move on now to the second tool that we used in multivariate analysis, which is called partial least square, PLS in short. So now we will have on one side the predictors, which are the spectral data, 800 or more than 800 columns. On the other side, we have the dependent variables, which are the component levels. And in between, what we want to do is to create a model that we call a PLS model that will be able to predict the composition from unknown spectra. We will use first derivative and the 240 to 300 nanometer ranges. So PLS, unlike the PCA, is called a supervised method. Why? Because we are putting in the model while we are training it both the spectral data and the component. So we are putting a target in a way. It is also based on reduction of dimensionality. So we will create latent variable. This time we will call them factors instead of principal component, but the idea remains exactly the same. It's to be able to summarize the variation we have in the data into a lower number of variables. And finally, as it is a regression, we can make some prediction with that kind of models. For the model training, we will combine calibration set number 1 and number 2 spectra, and we will use as an output variable the known sample composition, and we will train the model. This is what we get as a score plot for this exercise, for this calibration. Actually, we see something that is very close to what we have seen with PCA. The sample are cluster reflecting the sample composition. We have grouped replicates except for the outlier replicate, which is still in blue on this graph. Okay. Nevertheless, even if we have this outlier included in our data, we will try to see how good this model can perform. And to evaluate its performance, we will be using the validation set. Remember, the validation set has been created or prepared with freshly prepared stock solution, eventually using new batches for the API and for the preservative. And that's why we are presenting to the model something it has never seen in the past. And we also know the composition, but of course we will not give it to the model. We will try to predict the value. This is what we get for the API, where we are plotting here the predicted value against the reference value. In blue, you have the calibration sample, and in red, you have the validation samples. What we see is that the predicted concentration is close to the actual concentration. We have an error, an RMSE of about 1.7%. And the samples line up gently with the target line that we have here. So that's good. And as you see also, the validation sample are falling close to where they should be. The prediction are falling close to where they should be. On the other hand, for the preservative, we actually see that the predicted concentration is close to the actual, except for the outlier samples, which are here, 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 and here. Okay, and of course, the RMSE will be much, much higher, around 5%. The reason is because we have these outlier values. Notice that the validation, despite the presence of this of these outlier set, the validation samples are actually mapped to the right position. So it's pretty encouraging that we see this. Okay, these are the outlier samples I have highlighted. Let's try to rebuild the model, but excluding this outlier sample set. And we recreate the PLS regression. This is what we get, and it's very similar to what we have seen in PCA. The sample will cluster reflecting the sample composition. We have grouped replicates, and the location of calibration sample is very comparable to the DOE structure that we had created initially. In terms of validation of the model, the input variable will not change. These are the validation set spectra, and we know the composition of these samples, and we will compare prediction to the actual values. This is what we get for the API. We have slightly improved the model. So excluding the outlier from the calibration set has slightly improved the RMSE, so the model error. The validation sample, as we have seen previously, are very close to between predicted and actual value. But the real difference that we see is now visible on the preservative. So here we reduced the error of the model from 5% to less than 1%. So this is a great improvement in the quality of the model. And we have validation samples that fall really close to where they should be. So to summarize the findings we have from this parcel least square regression, PLS was applied where the inputs are the spectral data and the outputs are the sample composition. The PLS allowed us to predict the composition of validation sample based on their spectra. And this is exactly what I have been showing in this corner of the slide. We have also identified an outlier calibration replicate in the dataset, which is most likely due to an error with the preservative concentration. As you have seen, if we include the outlier, we have a very bad prediction capability regarding the preservative. However, for the API, actually including or excluding the outlier sample set has a slight effect compared to the preservative concentration. So that's why I'm saying here that it's most likely due to an error with the preservative concentration in the sample that we have that explain this outlier. As a conclusion, this method that we have developed is very quick, and it is very quick because measuring a spectrum, in this case UV-Vis spectrum, will take 1 to 2 seconds, and the model that we have created, either PCA or PLS, can be applied on the fly. They can be applied in real time, and thereby this actually fulfills the needs we had, which was about having a process feedback loop to be able to adjust the feed in API. Another advantage of this method is that you have seen we can assay simultaneously 2 ingredients in the solution without making any separation, so without applying a chromatography technique. Combining design of experiment with multivariate analysis was really a key success factor in this case. The DOE helped us to select which sample to test, which sample to put in the calibration, and eventually which samples to put into the validation. Why? Because design of experiment are good at covering the design space, as selecting which sample to test. Actually, the case that I've presented is quite easy. We had only 2 variables to deal with. Imagine if you had more variables, more ingredients to predict into the solution, 3, 4, 5, eventually many others. Eventually, you can also include some process parameter and constraint. So it becomes very efficient to work with design of experiment approach to have a very good coverage of the design space and select the minimum number of runs or minimum number of combination to do in order to get a valid calibration later. And for MVA, we have seen that we have used 2 tools, principal component analysis and PLS, a partial least square. PCA is a very useful tool if you want to explore the spectroscopic data and identify outliers. This is, in my opinion, a key factor, a key success factor to do before jumping into the regression, the actual regression. It's always good to look at the data before doing these type of regression. The PLS is an efficient way to build regression when the input data are correlated, because it will actually recognize these correlations and combine them, summarize them into the latent variable that we have been discussing, and thereby making the model much more robust and much more useful. To finish with, MVA model quality is very, I should insist on that, very dependent on the quality of the calibration set. And that's why the design of experiment is very important to use to define that calibration set. The presence of outliers can severely affect the model quality, and therefore they need to be investigated to have the, let's say, the best performing calibration set. Okay, I thank you very much for your attention, and if you have, I hope you have been interested in this presentation, and if you have questions, please let me know.
Okay, thanks, Sam. Oh, let's see, I'm going to get into sharing my screen now. I'm going to cover a couple of housekeeping details before we get to questions, so go ahead and type your questions up. First of all, the slides from this presentation have now been posted onto our website at staties.com/webinars. And let's see, the recording of the presentation will also be posted in a few hours after the processing of the video file is finished. But in the meantime, one thing that we are doing for attendees to this summit is we're offering a discount on our distance learning workshops. And you can go to staties.com/training/live for a list of upcoming workshops. This information will also be sent in the follow-up email that will be sent out to you tomorrow, listing. Also, it'll list where to find the slides and the recording. I should say that that discount code listed right there is valid until August 31st for courses attended by December 30th. Okay, now let's get into some questions here. Let me bring up the questions area. And let's take a look here. Let's see. Okay, Sagar Suman Panda asks, can we apply these principles of PCA and PLS and LC and FTIR2? Secondly, what new findings will we obtain by applying these approaches?
Yeah, I think these algorithms that I have been describing, PCA and PLS, can absolutely be applied to infrared spectroscopy, including FTIR. We applied them successfully also to NIR to determine the concentration of an API in a solid dosage form. So that's another application we have conducted with this. And yeah, the learnings that we can get from applying this is it's actually similar to what I have been describing during the presentation, is that we summarize the information that we have in the spectra, which is often correlated between the wavelengths, into a neater set of variables that we can manipulate more easily. So that's about the dimensionality reduction that that I described earlier. Moreover, I should say that applying PCA or PLS is not only limited to spectroscopy. We could also apply it to process data, where also we could have some correlation between variables that we measure at different points in the process, and actually using PCA and PLS will recognize these correlation and bring some neater view about what is happening in the process.
Okay, great. Okay, let's see. We have Manpreet Bhatti asks, is it simple PCA or Varimax rotation applied? How many columns and rows were there? And factor analysis based on scree plot can give better results. Any comments?
Can you repeat the last part of the question?
Let's see. Factor analysis based on scree plot can give better results. Any comments?
Okay, I have to admit I don't know this technique. What I can say is that PCA is a matrix factorization method. What we have applied is regular PCA using either SVD decomposition or it's really regular PCA that you can find in textbooks.
Okay. Let's see. We have a question from Omer Gull. How did you transfer UV visual spectra data into StatEase?
Actually, I never needed to transfer the data into Design Expert. What I tried to highlight in the presentation is that I've used Design Expert to select which combination of concentration of API and preservative I should run into the calibration. But actually, I'm not transferring any information to Design Expert later. I'm using exclusively Unscrambler to do the analysis.
Okay. T. Beals says, very nice work, thank you. Did you do any HPLC to assess/confirm the spectroscopy results?
Yeah.
For a given sample, API preservative, how do you HPLC and spectroscopy accuracy and precision compare?
Yeah, good question. We did some preliminary testing. So that means we have taken, for example, samples that are on target, which are samples from commercial products, for example, that we test HPLC because we need it anyway for the release, and we test them as well for using the UV-Vis chemometric method, and the agreement was quite good. Although I should recognize this is not a full validation of the method. We will need to do some more work to prove, to document that these two methods are equivalent over time.
Okay. And Morad Malal asks, how many HPLC columns for this experiment, as we know that these bring a lot of variability on the measurements?
Mm-hmm. Okay. Well, in this case, we have a method that is quite established for a long time now. The influence of the column has been evaluated over time. So, and as I mentioned, actually, I'm not using as such the results from the HPLC. I'm really creating samples with by weighing, if you want, combining known amount of API and known amount of preservative by weighing to prepare my calibration samples, and I'm not using the HPLC as a comparison.
Okay. Let's see. Next, Anastasis Mitsopoulos asks, MVA puts data in a plot, but are there no indicators that let you better interpretate the data? What if the results create no clusters that are symmetrical?
That's a good point. Actually, we were hoping to see the pattern that I've been showing, as you remember, maybe the first plot that I've shown was not showing at all the pattern I was expecting. So that's why using MVA is also a good way to understand or to nail down which pretreatment and which wavelength ranges are the most important or the most representative of the data that I want to cluster together. So yeah, using PCA as a naive method, which looks only to spectral data and just playing with the pretreatment, playing with the ranges of wavelengths is a good way to actually have a sense of what do I need to do to the data to be able later on to make a good regression model. So this is how we used it. I hope this answered the question.
Okay. I think we have time for one more. Let's see, this is from Ruben Murcia. And he writes, I guess Design Expert can also be used to get the regression and hence be able to predict as you did with Unscrambler software. Why did not use Design Expert then? I guess he's asking why you went the direction you did.
Yeah, actually, Design Expert will unfortunately not be as such able to build the same models that I have created. if you recall in the presentation, the first step is to reduce from 800 columns to something like 2 columns, which are the principal component. So, and this part of the work is not actually feasible in Design Expert. There is no algorithm implemented in Design Expert to do that. Therefore, we need to do it in another software. So that's why I have not used Design Expert for this explicit calculation. What you could do eventually is, once you have created the principal component and you have the score plots of each sample, you could actually use these scores on each principal component to be predictors of the composition, and this can be done in Design Expert. But for me, I see it like a bit more cumbersome to go back and forth between softwares.
Gotcha. Okay. Well, perfect. Well, thank you very much, Sam. We appreciate the presentation.
Thank you.
And thank you, everybody, for stopping by, and we'll see you again on a future webinar.
Thanks.
Awesome.
Thanks a lot.
Thank you. Thanks, everyone. Take care now.
Bye.
Bye-bye.